Good morning, students. Today we are studying about the plant tissue culture. Plant tissue culture. Here show the some endangered plant species Rahulfia, Banda and Mangifera. Plant tissue culture. Plant tissue culture means the regeneration of entire plant from a single tissues. That is called as a plant tissue cultures. The plant tissue culture is grown under the sterile conditions on a nutrient culture medium. The plant tissue culture is widely used to produce the clones of the plant in a method known as micropropagations. The advantages of tissue culture, it is conservation of the endangered species and the creation of large number of clones using a single explant, selection of and culturing of desirable traits and reproduction without having a wait for production of seeds and short production time of species with long generation times, no seasonal restriction on plant germination. Techniques of plant tissue culture. First is of glasswares, preparation of medium, explant preparation, inoculation, shoot formation, root formation, organogenesis, and first one is the nutrient media preparations. There are so many nutrient mediums for tissue cultures. Here mentioned for Murashi-Janskog medium. It's discovered for 1962. Murashi-Janskog. Here there are many micronutrients, macronutrients, carbon and energy sources are present in this nutrient medium. Vitamins, amino acids and other inorganic salts. 1 to 3% of sucrose and solidifying agent. In the solidifying agent we used for agar-agar. 0.8% of agar used for solidifying agent, and the pH medium. pH medium is mainly checked for the plant. Next one is plant growth regulators. The plant growth regulators or phytocarbon is important for the promote the growth, development and differentiation of plants. 4 broad classes of growth regulators. One is auxin, second cytokinin, third gibberellins, and fourth abscisic acids. What are the main functions of plant growth regulators? First, promote the growth, differentiation, organogenesis of plant tissue cultures. Next, the 4 growth regulators. What's the main functions of growth regulators? For example, auxins, it is induce the cell division, cell elongation and formation of callus in cultures, and high concentration of auxin initiate the root formations. Next one is the cytokinins. What's the main function mean? It promotes the cell divisions, root shoot formations and RNA synthesis and thus stimulate the protein and enzyme activities in tissues. Next one is the sterilization of glasswares and culture medium. It's very important for plant tissue culture. This medium is sterilized under the 15 lbs and temperature at 121 degrees Celsius for 15 to 20 minutes. The sterilization is important to avoid the bacterial contaminations. The medium is poured into the culture bottles and these bottles are again steriled in the autoclave. Next one is the selection of mother plant. It's very important for tissue culture because the sterilization of sterile plant is important for tissue culture. The sterilization accomplished for 70% of ethyl alcohol for 60 seconds. Next, inoculation. Inoculation of explant on sterilized medium. We have to cut the explant and steriled under the humidity chamber used for a root, shoot or any part of the plant. After 2 or 3 weeks, the plant callus is growing on the plant tissue culture. What is the callus formation means? That is undifferentiated mass of tissues. In the living plant, callus cells are those cells that cover a plant. Then shifting of cultures for subculture. After 3 to 6 weeks from inoculation, the inoculated explant shows growth in multiple shoots. These shoots are transplanted or subculture or multiplication medium for further growth of plants. For subculture, separate growth medium is prepared. Several subculture cycles are done in the laboratory for mass production from the Then separation of in vitro shoots and rooting. The shoots are separated in their aminar airflow chambers and fully grown with the shoot, then transferred into the rooting medium for root generation. Depending upon the species, rooting requires for 1 or 2, 3 or after complete growth of the plants, the plant should be hardening. Hardening means the healthy plantlets are exposed to the natural condition. That is mean is hardening. When the polythene cover is completely closed for the new explant growing. The plants are kept in hardening room with controlled condition of light, humidity and temperature are maintained. After complete the growth of plants, the plant will be transferred for greenhouse. The greenhouse effect is a house with the production of boundaries which allows only and sunlight and no other outside environmental factor to enter. It provide and maintain a growing environment that will result in optimum crop production at maximum. I think you understand for plant tissue culture. The plant tissue culture is mainly used for propagation of endangered species and multiplication of the species, and it is production of disease resistant varieties and so many uses coming under for plant tissue culture. So you can understand, I think. So it's the plant tissue culture techniques and their uses. Thank you, students.